Hello everyone. In this chapter of Life on Planet Earth, we are going to focus our attention on the patterns of inheritance. Basically, why do you look the way you look, right? Now, as we dive in this chapter, I want you to keep in mind what we covered in the previous chapter. We talked about meiosis, and we talked about meiosis being the engine for genetic diversity in the population. We talked about how genes are exchanged, how chromosomes are shuffled during the process of the formation of gametes, whether it is sperms or eggs. The process applies to both, right? Now we are going to take a look at a more practical view of the role of meiosis in the patterns of inheritance. With that being said, our first stop is going to be on talking about Mendel, who is actually one of the fathers of genetics. Mendel did a tremendous amount of work that laid the ground for modern genetics. Now, Mendel actually was born in 1822 and died in 1884. Now, that's not the detail I want you to remember, though. That's not the detail I want you to remember. What I want you to remember, it's the work he did and the conclusions of that work he did. Now, Mendel worked with garden peas. As he worked with garden peas, he was interested in what garden peas looked like and what was actually hiding behind the physical look. For instance, some of the things he observed, and actually he called them traits, right? Some of the traits he looked at was, for instance, the length of the stems. Some stems were short, others were tall. He looked at the pods. Some pods were inflated, some were constricted. He looked at not only the color of the seeds, but also the shape of the seeds. So some seeds were wrinkled, others were round, some were green, others were yellow. He looked at the position of the flower on the stem. Were they axial or terminal? Also, the color of the flowers, were they white or purple? And finally, he looked at the color of the pods, were they yellow or green? So for all these traits, Mendel had performed this work that allowed him to reach that, well, all observable traits have something behind that he called factors, right? And these factors actually can be expressed or not. It just depends based on its observations, right? Now, this is what we are going to dive in, but how did he go about studying these different traits? Well, working with garden peas, here we have the flower, the typical flower of a garden pea. Now, you have the stigma, which is actually one part of the reproductive female organs in plants, and you have, on the other hand, the anthers that are yellow here. These anthers actually are going to house in the pollen. When they open, they release pollen, right? While the egg is going to be right at the base of the structure of the plant, right? Going into the stigma, going very much down, the egg is going to be down here, okay? So what Mendel did is to try to isolate some reproductive organs so that he can cross-fertilize plants. In other words, one plant is not going to fertilize itself, but he's going to intentionally play the role of a pollinator, right? He's going to go take a brush from one plant, take a brush and obtain pollen from one flower, and then on the stigma, he's going to actually apply pollen on the stigma from a different plant so that intentionally he can look at, okay, well, if I want to take a look, for instance, at the color of the flowers, one plant has white flowers, the other plant has purple flowers. Well, he's going to cut the anthers so that, for instance, the purple flower do not fertilize purple flowers, right? But from the white flower, he's going to obtain the pollen from the white flowers and bring them to the purple flowers and vice versa, right? So this is what he worked on. And very importantly, the reason why Mendel actually did this work on pea plants, it's because the pea plants are relatively easy to work with. They have a short life cycle. They reproduce many times during the year, right? So he was able to control the environment so that within one single year, he had multiple reproduction events and was able to run some math, because he didn't just do the pollination and collected all these features, but he also did a lot of math so that he can come up with some prediction as to what he was doing. Now, the first aspect we are going to take a look at is the one-trait inheritance. What we mean by one-trait inheritance, it's that Mendel could look at, say, the color of the seeds. That alone would constitute one trait. He could look at how seeds were shaped. Were they wrinkled or round? Just looking at that would constitute one trait. Now, in this very example, we are going to take a look at the color of the pods, right? Some pods are green, others are yellow. Now, Mendel always started his breeding with what we call true breeding. The true breeding are this breeding that when breeding the plant from one generation to another to the next, the feature he was targeting was exactly the same. No variation, right? For instance, a true breeding for green pods would produce green pods all the time from generation 1 to generation 2 to generation 3, 4, 5, and so on. That would be a true breeding for that feature. And he would do the same thing for the color yellow. Now, I'm anticipating here a little bit. You have the physical trait that we see here, the color. And then you also have another aspect here, capital G, represented 2 times, and lowercase g right? Represented two times. So this capital G, and lowercase g represent actually the factor that explains why the color of the pods are green or yellow. So that represent also one G coming from one parent plant and another G coming from the second parent plant, right? This is also true for the lowercase g. Now the question is going to be, considering that genetic makeup, right? Remember during meiosis, we have homologous chromosomes, right? These homologous chromosomes are going to carry the same genes at the same location, even though these genes can be present in alternate forms, right? We are going to address that a little later. So now the question is, from this genetic makeup that we also call genotype, right? What would be the gametes produced by that individual? So because capital G is represented twice, regardless of how we turn things, the gametes are only going to produce what kind of gamete? It's the gamete with capital G right here. Likewise, when we look at the plant with the yellow pods, they're going to produce one kind of gametes, the gamete with lowercase g. So then Mendel brought these two gametes together, and what he observed in the first generation, he observed that all pods were green. That observation caused Mendel to think that, it looks like there is one factor that is actually expressing while the other is not expressing, right? We are going to come to the specific terminology he came up with at some point. So he observed that in the first generation, all single individual plants carried green pods. For that reason, because he started with two breeding plants that were different, he called the first generation monohybrid. Now, when crossing the monohybrids, the question is, that monohybrid, what kind of gametes are they going to produce? They're going to produce, some gametes are going to carry capital G, other gametes are going to carry lowercase g, right? So now, considering that, if you consider an egg carrying capital G and a sperm carrying capital G, when a sperm fertilizes the egg, we obtain a plant that is going to be capital G, capital G. By the same token, if the sperm with capital G was to fertilize an egg that is lowercase g, then we would have a hybrid individual that is capital G, lowercase g, right? The color of the pod would be green. And when considering the sperm with lowercase g, crossing or fertilizing an egg that is capital G, the individual is going to have pods that are capital G, lowercase g. The color would be green. And finally, when a sperm, lowercase g, was to encounter an egg that is lowercase g, that would produce an individual that is lowercase g, right? And the pod would be yellow. So Mendel, doing some math, was able to come up with some prediction. Now, in reality, in the work I'm showing here, there is a mix of what Mendel did and some other people did, including a guy called Punnett. right? And actually what you see right here, it's what we call the Punnett square. But I'm not going to bug you with that detail. What I want you to basically focus on, it's actually how we come up with the genetic makeup of the individual, considering the genes that the sperm carries and the genes that the egg carries, right? What would be the genetic makeup of the outcome, of the offspring, and what would be the observable trait of that offspring, right? So in terms of observable traits, we see that 3 individuals out of 4 are green. So that represents 3 out of 4 green, and 1 out of 4 yellow. So we have a ratio in terms of the color of the pods, green to yellow, it's 3:1, right? 4 being the total possible, right? So that it's actually the ratio of the phenotype. Phenotype, it's the observable traits, while genotype, it's the genetic makeup. Please remember that. So we have genotype, right, which is a genetic makeup. Genetic makeup, all right? The phenotype is the observable traits, right? Capital G, again, being green, accounting for green, lowercase g accounting for yellow. So the conclusions of what now we know as the first law of inheritance is the law of segregation. That law of segregation says that each individual inherits some genetic information from mom and some genetic information from dad, right? Homologous chromosomes. Now, when these homologous chromosomes separate during meiosis, the allele you receive from mom and the allele you receive from dad end up in 2 separate cells. Regardless of how we put things, that's what is happening, unless there is an event that we called during the previous chapter non-disjunction event, right? But in as much as things are happening properly, the 2 alleles are going to end up in 2 separate cells, alleles being alternate forms of the same genes, right? So each individual has 2 factors, factors being genes, right? For each trait, okay? Trait being the phenotype, right? The factors segregate, so they separate during the formation of gametes, which we know to be meiosis. And each gamete contains only 1 factor, okay, or 1 allele for each pair of factors, okay? Fertilization is going to give back the possibility to reconstitute the genetic makeup, right? Now, sometimes Mendel would know the genetic makeup of one plant, but wouldn't know the genetic makeup of another plant. So then he wondered, how can I tell the genetic makeup of the unknown plant? So what he did, he was actually to work with the yellow pods and the green pods. So the yellow pod, he knew that they were lowercase g, no question about it. The green pods, not too sure. were there capital G, or capital G, lowercase g, right? So from that perspective, he drew hypothesis, right? If crossing yellow and green, and that in the offspring, he observes a ratio of 50% of individuals that are green and 50% of individuals that are yellow, he would come to the conclusion that, well, the parent has to be a mix, right? A monohybrid. While on the other hand, if he cross again yellow and green and observes that all the individual, 100% of the individual produced green pods, meaning that the parent up here was necessarily carrying capital G. I'm trying to avoid the use of the correct term, which actually, let me say it now, it's homozygous dominant, right? Homozygous dominant. We are going to see that in a minute. So again, the one thread test cross allows us to say, knowing one of the parent's genotype, to determine what the genotype of the unknown parent would be, okay, by way of two hypotheses. All right, so now let's dive into the modern look at Mendel's work. Now remember, we have two sets of chromosomes that are homologous chromosomes, right? One we receive from mom and one we receive from dad. And each set of chromosome is going to have genes that are at different location. The location where a gene is found is called a locus. I want you to remember that. One locus, several loci. Loci, it's basically multiple locus, right? Loci, okay? So now, the term trait actually was the gene, okay, that eventually translate into phenotype at some point. And then the term factor was the alleles being alternate forms of the same genes, right? So we have, for instance, for capital G here, lowercase g. On the other hand, these are two alternate forms of the same gene. If we have capital R here, we have lowercase r here, and so on. So now, during meiosis, we are going to have the duplication, first of all, of the genetic information. That duplication of the genetic information is going to cause sister chromatids here held together through centromeres, right? This is true for the chromosome you receive from mom. This is also true for the chromosome you receive from dad, right? So now remember that these chromosomes are going to undergo division, right? The division of the nucleus and distribution of the chromosomes during meiosis, right? Hence, the law of segregation, which is that the homologous chromosomes, the sister chromatids are going to separate, right? So the genotype, it's the combination of alleles in the cell of an individual, a diploid individual, right? So that we are going to have homozygous, which is two individual alleles that are exactly the same, right? And then we're going to have the heterozygous individuals, which is that individuals are made up of two alleles that are different. So two different alleles. Then we have the phenotype, which is the physical appearance of an individual, what we can actually see. We cannot see the genotype, but we can see the phenotype, right? For instance, here, now, when we look at allele combination, if an individual is capital A, that means that this individual is homozygous dominant. Now, in the basic genetics, we always represent the gene that is expressing, the allele that is expressing with a capital letter. While the allele that is not expressing, we always represent it with a lowercase letter. Please note that. So, when we look at the pigments, for instance, if capital A is presence of pigment, lowercase a is no pigment, right? This individual capital A, homozygous dominant, is going to have presence of pigment, right? Normal pigmentation. The individual who is capital A, lowercase a, that's the genotype, right? Is going to be heterozygous. Hetero because there is a mix of a dominant and recessive, right? That individual, in terms of phenotype, is going to have a normal pigmentation. However, an individual who is going to be homozygous recessive, right? This individual is going to have no pigmentation, so that would be the case of albinism.